Dear students, the topic is immobilization of enzyme.
Before going to the topic, please subscribe my channel, press the bell icon for notifications, like, share among your friends and comment.
Enzymes are biocatalysts and have wide application in industries. Enzyme produced by microorganism can be used for the production of various industrial products. The term immobilization of enzyme means adhering an enzyme on a surface is called as immobilization of enzymes. Advantages and disadvantages for this process. Advantages, enzyme will get in pure form and can be used repeatedly for the production of commercial substances. When we examine the disadvantage, it is a time consuming process and costlier. The advantages of enzyme immobilization are the reaction is very stereospecific, that is the enzyme will attach the function group at the proper location. The final product obtained is in a pure form and the one formed by the synthetic process is racemic. The yield obtained is more compared to chemical synthesis. Enzymatic reactions are carried out in mild conditions such as low temperature and less toxic chemicals. Enzyme can be reused whereas chemicals can't be reused. Eco-friendly reactions if the enzymes are immobilized. The disadvantages are time consuming, makes the drug costlier. Sometimes there are errors in the purification, isolation, and the inactive toxic form remains in the final product and cause serious side effects. Next we can see various methods used for the immobilization of enzymes. There are different methods used like adsorption, covalent binding, entrapment and micro encapsulation. Have a look on these methods in detail. These are the methods like adsorption, covalent binding, entrapment and micro encapsulation. Adsorption. In this technique, the enzymes are adsorbed onto a polymetric matrix. Adsorption occurs due to electrostatic, hydrophobic or affinity binding. During adsorption, pH should be controlled carefully because the change in pH may lead to desorption. That means detachment of the enzyme. If the enzyme is released from the substrate, it gets mixed with the product and cannot be reused. Also, a purification step is then required. Some of the common examples of adsorbents are alumina, bentonite, CMC that is carboxymethyl cellulose, CMS silica gel, DEAE cellulose, dimethyl aminoethyl ion exchange resins. See in this diagram, it is shown that is the attachment of enzyme on the surface of the substrate. Covalent binding. The amino groups on the enzyme form a covalent bond with the active groups on polymer support which is used to immobilize the enzymes. This can be done by two ways. Through the reactive groups on the side chains of its amino acids such as lysine, arginine and tyrosine. Through the terminal amino and carboxyl group of polypeptide chains. Carrier matrix used are polyurethane, CM cephadex, porous glass. The enzyme covalently bind with the substrate. Enzyme entrapment. Enzymes are entrapped within the matrix or gelatin and cannot escape permeation. Enzymes are entrapped in such a way that only product and substrate can diffuse through the pores of the matrix. The substrate enters the matrix, react with the enzyme and then the final product diffuses out of the matrix. So pure products can be obtained. The enzymes and the polymeric substance remain immobilized. Commonly used polymers are gelatin, agar, starch, etc. See in this figure, entrapment of enzyme on the gelatin matrix. Next is microencapsulation or membrane confinement. The enzyme are confined into the capsule. The capsules are made of permanent material like nylon, collodion, etc. Biodegradable materials like polylactic acid or phospholipid liposomes are also used. These are pores in the capsule. The pore size is adjusted by changing the concentration of ingredients used for manufacturing of capsules. Through these pores, the substrate can enter inside the capsule and interact with the enzyme, and the product will be formed. The product will come out through the pores of the capsule. See here, enzymes are encapsulated, then porous membrane in the figure. Next we can see applications of enzyme immobilization. Here is the application: biosensors, electrochemical sensors, glucose sensors, etc. Enzyme reactors, batch reactors, continuous flow reactors. Application of immunoadsorption technique, immobilized enzyme based immunoassay, immobilization based novel drug delivery system. Analytical, immobilization in affinity purification, affinity chromatography and purification. Medical, detoxification following a drug overdose. Pharmaceutical, selective hydrolysis of penicillin G, steroid modification, production of monoclonal antibodies, animal vaccines, etc. Dear students, I hope you know what is enzyme immobilization and its applications. Thank you for watching. See you in next class with another topic. Don't forget to leave a like and please subscribe my channel.